Hemophilia is a genetic disease in which factor IX of the clotting cascade is defective. This leads to impaired coagulation. Patients experience bleeding episodes throughout their lives. They suffer chronic joint damage and early mortality. There are three levels of severity. The mild form of the disease, in which patients have 5 to 40% of the normal level of factor IX, moderate severity, in which they have 1 to 5%, and severe disease, in which they have less than 1% of the normal level of factor IX. The severe form of the disease is the topic of our talk today. Hemophilia is a rare disease with a prevalence at birth of 5 in 100,000 males. About a third of these cases have the severe form. In high-income countries, severe hemophilia patients are diagnosed within the first few months of their lives. However, in low and middle-income countries, many patients die early or are never diagnosed. There are several treatment options for hemophilia patients. Patients can receive on-demand fresh frozen plasma to control bleeding episodes when they occur. Fresh frozen plasma is a blood product accessible in most blood banks. However, this option comes with the risk of volume overload and blood-borne diseases. The second option is to infuse patients with manufactured factor IX to control bleeds once they occur. In this case, factor IX should ideally be infused in standard dose, but if not affordable, it can be infused in low dose instead. A better option is to prophylactically infuse patients with factor IX on a regular basis, which is about 2 to 3 times a week. This reduces the occurrence of bleeding episodes and minimizes joint damage. Again, ideally prophylaxis should be provided in the standard dose, but if not affordable, then it can be infused in low dose protocol. In high-income countries, the standard of treatment is standard dose prophylaxis. However, in most low and middle income countries, this is not an option, and they have to resort to cheaper, less effective options. Lastly, gene therapy is a new treatment under development and FDA approval. It promises long-term results after one infusion. So in the previous slide, we talked about how a big proportion of hemophilia patients never got diagnosed in low and middle income countries. But even if they did get a diagnosis, do they have access to treatment? The pie chart on the right shows the distribution of factor IX in different geographical regions. For example, Africa receives less than 1% of the global factor IX distribution. Gene therapy is a one-time infusion, and it can transform lives of many patients in low and middle income countries who suffer a very poor quality of life due to difficulty to access factor IX. However, this might be wishful thinking if gene therapy is priced at $2 to $3 million, as has been announced in the gray literature, making it the most expensive drug in the world. Last year, our group published a paper in Blood Journal in which we disclosed the manufacturing cost of one dose of gene therapy as retrieved from the GMP facility that produces the vector in St. Jude Hospital. The total cost, excluding research and development, was less than $80,000 per patient. This highlights the massive gap between the manufacturing cost and the suggested price. St. Jude is currently moderating a hemophilia gene therapy clinical trial in which 8 sites in 6 low and middle income countries have been identified to participate. Costing and cost-effectiveness analyses are being conducted as part of the ongoing clinical trial. Therefore, our objective was to evaluate the lifetime cost and cost-effectiveness of comparing gene therapy versus all and any available alternative treatments in severe hemophilia B patients in 6 low and middle income countries using real-world data to explore a country-specific equitable price point. By real-world data, we mean an on-the-ground dosing protocols, unit costs received from hospital records that our partners were able to provide us. We constructed a microsimulation Markov model with weekly cycle and extending over a birth till death time horizon. All patients were born in the no inhibitor health state, and then they could experience one of 3 complications: minor bleeds, major bleeds, and joint damage. Once joint damage developed, it lasted for many years until patients undergo total joint replacement surgery. These surgeries were not affordable and not an option in most of the sites we explored. Major bleeds were accompanied by the risk of death. We also accounted for background mortality at any stage of the cycle. Patients could develop inhibitors against the external factor IX. This complicates the treatment because patients in this case stop being responsive to factor IX and need an alternative, more expensive treatment, which was not available in any of the sites we studied, meaning that inhibitor patients who experience a bleed in these settings have high mortality rates. Finally, gene therapy was available, and the youngest age at which it can be infused at the moment is 18 years. Patients should be free of inhibitors to be eligible for gene therapy. And based on clinical trial data, we assume that once infused, no bleeds will occur for the first 9 years after infusion, and then incremental minor bleeds will occur between years 9 and 31, and then after 31, patients would be switched back to on-demand or prophylaxis. Here we discuss the sources we used to retrieve the model parameters. So dosing protocols and costing data were collecting through an online survey, and then virtual meetings were set with a team in each of the participating sites to validate and double-check some information. We used country-specific published average male weight in every year of life, and we used the suggested $2 million price point for gene therapy. Then for utilities and transition probabilities, we retrieved these values from the literature, and the microsimulation model allowed us to create ranges whenever needed. Finally, for mortality, we used country-specific mortality rates published by the United Nations, and then multiplied it by risk rate specific to hemophilia patients. As a first step, here we display the costing results. Factor IX was not available in 3 out of the 8 sites, therefore prophylaxis was not an option. For the remaining 5, a proportion of the patients received low-dose prophylaxis, while only in Lima, Peru, half the patients received standard dose prophylaxis. When patients experienced bleeds, only 3 sites out of the 8 could offer them standard dose factor IX concentrate. Prices varied widely between sites. Factor IX unit factor cost ranged from 0.3 to 0.45 US dollars. Fresh frozen plasma ranged from $7 to $24. Then when we calculated the factor cost for a whole year of prophylaxis, we found that it ranged between $6,000 to $54,000. A minor bleed treatment ranged from $60 to $4,000, and major bleed ranged from $420 to nearly $16,000. Lima, Peru was often at the highest end of the range due to more intense treatment protocols, high unit cost, and higher average weight of patients compared to the other countries. In this table, we can see the results of the cost-effectiveness analysis. The x-axis shows the sites studied, and the y-axis shows 5 treatment approaches arranged from the least to the most intense and expensive. For each treatment approach, we combine gene therapy, which is only effective from 18 to 50 years of age, and thus, before and after this period, patient receives on-demand or prophylaxis. We assume a threshold of $100,000 per quali, which is a high-income country threshold level and not feasible in most low- and middle-income countries. Yet, assuming this high threshold, we found out that gene therapy was not cost-effective in most cases. In the first 3 sites, where only fresh frozen plasma was available, the lifetime cost ranged from $100,000 to $200,000, and accumulated qualis were less than 10. This is lifetime qualis. Gene therapy was not cost-effective in any of these sites. The next 4 sites offered on-demand and prophylaxis factor IX concentrate. Costs ranged from $350,000 to $1.3 million, and qualis ranged from 11 to 17, and still gene therapy was not cost-effective. In the very last site, which is Lima, Peru, gene therapy was not cost-effective when compared to standard dose on-demand, but was dominant when compared to standard dose prophylaxis. Our results show that the ICER was never below $116,000. This is similar to the massive ranges that we saw in the previous slide. Here we see that the lifetime cost of a patient in Davao is $100,000 and that of a patient in Lima is $2.3 million. So what is the suitable price point? For this exercise, we explored the point at which gene therapy becomes dominant, meaning better effects and fewer costs, compared to the alternative. We opted for the dominance threshold because in many countries there is no preset country-specific threshold, cost-effectiveness threshold. And also in the grand scheme of things, the margin between cost-effectiveness and dominance is minute and it would not affect the price significantly. Again, we can see a massive variation across countries and between different cities in the same country. While gene therapy manufacturing cost is about $80,000 and proposed price is $2 million, the equitable price point for the 8 sites ranged between $50,000 and $2.1 million. So, circling back to the equity question, it is notable that the most deprived sites where gene therapy can increase qualities by 50% are the sites that need gene therapy to be priced the lowest. To summarize, collecting real-world costing and cost-effectiveness data in parallel with clinical trials is vital to assure the feasibility and realistic application of novel treatments in different markets. From an equity perspective, pricing decisions cannot be based solely on high-income country perspectives. Otherwise, that will increase pre-existing disparities. And finally, innovative pricing strategies and payment models should be tailored to account for differences in different low and middle income countries. Thank you for listening to my presentation, and thank you to all the sites that participated in this study. Thank you so much.